All right, so today we're going to talk about what you're going to do with all the stuff you've been working on lately. So, um, right, so last time what we've done is we made a PCR product. We digested it with a couple of restriction enzymes. We did some DNA cleanups. And so hopefully we'll have a PCR product that has some sticky ends hanging off of it. And again, we're going to call this an insert now. And so we did the same thing with the plasmid, and that has the complementary sticky ends, right? So you can see this AATT is going to base pair with that TTAA right there. Here's a GAT, CATG. It's going to base pair with that GTAC. Okay. Um, and so the next thing we need to do is really we bring these two things together. We have to make sure that we have our DNA quantified. And again, the numbers that you should be seeing from your DNA quantification is going to be really low compared to other stuff we've done this semester, and that's okay. So when we bring these two things together, they're going to do their base pairing. But the thing that's not going to happen automatically is the DNA backbone, right? Remember how we say the DNA backbone is a chain of covalently linked um, ribose and phosphate or deoxyribose and phosphate. And so that covalent chain is broken at each of these sites. And so what we do is we add an enzyme called T4 DNA ligase, and that seals up the DNA backbone here. Okay. And so you can see now it's a covalently linked complete circle of DNA. The ligation reaction is very simple. It's basically a buffer, an enzyme, your two pieces of DNA, and then some water to bring the volume up to 10 microliters. And so this should seem really familiar to you, right? Everything that we do is going to have some DNA in it. It's going to have some buffer. It's going to have an enzyme. It's going to have some water. These are the standard kinds of reactions that we do in this lab. It's just what particular components go into it. Um, the thing about the ligation reaction is that we typically want a particular molar ratio of insert to vector. We generally want 3 pieces of insert for every 1 piece of vector. And so this equation that it's also in your protocol um, defines how to figure that out. Okay. And so if you take the nanograms of vector times the kilobase size of insert, divide by the kilobase size of vector, multiply this times that molar ratio, that gives you the nanograms of insert to add. Okay. And so we have certain fixed amounts that we use in this protocol. So we always use 25 nanograms of vector. Okay. So that goes right there. The kilobase size of vector is always going to be 4.7 kilobases. This molar ratio is going to be 3/1, so 3. The thing that's going to be different for each of you is the kilobase size of insert. Okay. And so, like, if you're working with VPAC2, that's going to be about 1.4. If you're using PCAP, that's going to be around 5 or 0.5. So, double check your kilobase size of insert, and then, plug all this stuff in there. That's going to tell you how many nanograms of insert that you're going to add to the reaction. Okay. Um, and so why do we have to do this, right? So we said before that plasmids are these little circular pieces of DNA that kind of float around inside of prokaryotes that are outside of their normal genome. The thing about plasmids is that in order for bacteria to utilize them, they have to be perfect. They have to be totally circularized pieces of DNA. Okay, so if we were to take just this piece of plasmid, right? It's a linear piece of DNA, right? It has this end and this end. It's not completely circularized. Bacteria cannot use that. Okay, so it doesn't do anything once they go in there. But if we add an insert, we seal up the backbone with T4 DNA ligase, then the bacteria can use it. The bacteria can't use this, right? Because this isn't covalently bound. These are only held together with the hydrogen bonds there. And so that's a very tenuous sort of connection. It's very weak, and so it basically falls apart very easily. And so bacteria cannot use this, but once we've sealed up the backbone, bacteria can use it because it's a nice circular piece of DNA. Okay. So we take that DNA and we add it to the bacteria. We call this transformation. Basically, any kind of prokaryote or single-celled eukaryote like yeast, if they take up DNA from their environment, that's called transformation. And so when we do it in a test tube and we force them to take up the DNA, that's also called transformation. Um, and so we use a technique called chemical competence. The bacteria that we buy them so that they're ready to take up DNA. Otherwise, you have to do a little bit of prep work. But if we do this into eukaryotic cells, so we're going to be working with mammalian cells in a couple of weeks, this is called transfection. And sometimes we can use a virus to force injection of genomic information into another type of cell, whether it's bacterial or human. That's called transduction. And so the third coronavirus vaccine is basically a transduction machinery to deliver a piece of DNA inside of our cells to get our cells to express a chunk of the virus DNA so that we can mount an immune response against it. Transduction is very useful for a lot of different techniques, but it should also be noted that the first two coronavirus vaccines that came out were essentially a transfection technique. They weren't transfecting DNA into the cells, but they were transfecting RNA into cells. And so this is also a very useful technique. So to talk more specifically about transformation, again, we just take a plasmid, we add it to a bacterial cell, and then we screen the bacteria to see if they have the right plasmid. Okay, and so we've mentioned before that our plasmid of interest has kanamycin resistance, right? And so what we do is we grow the bacteria on plates that have kanamycin in it. So theoretically, only bacteria that have taken up circularized plasmid will grow on this kanamycin media. How do we do that? There's two major types, chemical transformations and electrical transformations. We have a thing that we can use to do this. Typically, you have like a little a cell, it's called. You put your bacteria in there. Each side of the cell has metal plates, and you apply an electric field across the bacteria, typically in the range of 500 to 3,000 volts. That electrical charge causes holes to get poked in the bacteria, or this could also be eukaryotic transformation as well. Holes get poked in the cell membrane, the DNA can go in there, and then you can recover the bacteria after that. It's kind of a stressful situation, but typically they'll recover fine. Chemical transformation, essentially, we use a combination of osmotic shock, calcium, and temperature. So essentially, the bacteria, we kind of strip off their cell walls. Bacteria tend to have cell walls. Strip their cell walls off so that they're a little bit more fragile. We expose them to a little bit of an osmotic, so change in concentration of solutes around the organism, and a temperature shock. So the combination of those two encourages the bacteria to want to swell up a little bit. And in addition, we typically will have some calcium in there. Remember, our DNA has an overall negative charge. The calcium sort of counteracts the negative charge of the DNA to sort of make it more neutral, but also there's a lower calcium concentration inside the bacteria. So the calcium wants to go in, the DNA kind of interacts with the calcium, and the DNA goes in there as well. And so it's a way to sort of, we stress out the bacteria, they take up the DNA, and then we slowly get them to recover. So once we do this, again, we plate them on plates that have antibiotics, and we'll see colonies hopefully within a day or so. So what we can do is take the bacteria from those colonies and use PCR to screen whether the colonies have taken up the right DNA. Okay, so remember this PCR that we did a few weeks ago where we used the CMV forward primer and the SV40 reverse primer, right? And everybody did it against their plasmids. We saw products of about 821 bases. But remember what we're doing here. We've actually, hopefully, inserted a gene into this multiple cloning site. So whatever the size of that gene is going to add to the size of this plasmid. So the PCR product that we should see is going to be a lot bigger. Okay. So a lot of the times when we see colonies, sometimes it would have been a plasmid that has not taken up an insert. It would have just been an empty plasmid. And so we need to be able to tell whether we actually have our gene of interest in there or not. And so if we don't successfully insert a gene into the plasmid, then we'll see something at 821. If we have, then it'll be a lot bigger. So, if it's the CCAP, PCAP, it'll have an additional 500 bases. If it's the PAC VPAC2, it'll be an additional 1,400 bases. Okay, and so every PCR product or every colony that you screen will have a PCR product, right? Everyone will at least have something that's 821, but what we're really looking for is that bigger one. So hopefully, you'll see some down here that are 821. You'll see some that are a lot higher that would indicate that you've inserted your gene of interest. Okay. So that's our major work for this week. Once we have positive colonies, we make what's called a glycerol stock. You've all seen them. We grew the bacteria originally out of a glycerol stock. And when we have good bacteria that have our plasmid of interest, we want to freeze them down so that we can get to them whenever we need more of that plasmid. And so basically, it's a really simple protocol. We just add 850 microliters of the bacteria plus 150 microliters of glycerol. We vortex that really well. We assign each one with a number, and then we keep a database of all the things that we've cloned over time. And so then whenever we need that, we just go in the freezer and grow up some more overnight. All right. So for this week, right, you're going to take your two pieces of DNA that you have, you're going to ligate them together, you're going to do a transformation. This takes about 2 hours, but then you have to let the bacteria grow up overnight. Then you'll have colonies. You're going to do a PCR screen on them. Again, it takes about a half hour to set up a PCR. It takes about 2 hours to run, and then run the gel on that. Then once you have positive colonies, you're going to grow 6 mil cultures of them. You're going to use 850 microliters for the glycerol stock, and then you're going to use 3 mils for a mini prep. Okay, and so ideally you should have more than 2 micrograms of DNA, because that's what we're going to need for some of the work going forward. So if you get your yields up around 250 nanograms per microliter, that's generally pretty good. Anywhere over 200 should be fine. All right, have fun.